Welcome to Immunity Group's podcast, an educational podcast focused on translational science and global health. Today's topic for discussion is the resilience of the human gut microbiome and its stressors. We have David Spadaro joining us today. He is science communicator for the CSIRO Microbiome Research Group. Welcome to the podcast, David. Can you tell us more about what the Microbiome Research Group is currently doing?
And thanks for having me. The research is built on recent advances in what we call omics techniques, which are laboratory techniques such as genomics, metabolomics, transcriptomics, and proteomics. These analyses give researchers incredible insights into the communities of microbes, such as bacteria and fungi, and how they work together. It can tell us what species are present in the community and, more importantly, what they are doing and what role they are playing in the overall function of the community. By understanding this, researchers can predict and model the outcomes of the microbial community under different conditions, then take it further by intervening if they predict an outcome to be a negative one. They do this by changing out key members of the community so that the overall function can be improved. CSIRO's microbiome research team is using this approach to improve the health of wheat and cattle for food production, the breakdown of PFAS and plastics in the environment, improving the safety and quality of food in our supermarkets, and of course, our gut microbiome. Nina, I find it incredible how important the invisible but invaluable microbes are and how these tiny organisms can have such a large impact on our environment, food, and our health.
Well, this is the main reason for us having today as a special guest a gut microbiome researcher, Dr. Azadeh Safarci. Hello, Azadeh, and welcome to our podcast.
Hi, Nina and David. Thank you for having me here. So, hi everyone, the ones who are listening to this podcast. My name is Azadeh Safarci. I'm a research fellow at CSIRO and I'm a part of Microbiome for One System Health, calling it in CSIRO MOSH. So we do a whole range of different research on all types of microbiome, from environments to insects, animals and humans, and trying to find and investigate the interactions of these microbiomes together as one system health.
What a fascinating place to work at.
Thank you.
But what actually drew you into science? Like, what sparked your passion for the human microbiome research? Tell us, please, about your journey into this field through your experiences working with CSIRO, but also broader.
So let me start when I was in the high school. So in that age and in that era, so it was like introducing like all the biomolecular new approaches like PCRs, polymerase reactions, and primers, cloning, and all this stuff in our biology course. So I was pretty interested to continue these fields. And then I've started my undergrad in one of the high-ranked Iranian universities, Tehran University, in veterinary medicine. And then during my thesis, I've really fascinated to do some of these PCR methods, working on the different types of virulence factors in Escherichia coli, which is one of the most important bacteria which actually can be foodborne disease in humans as well. So we try to find out the virulence genes using PCR methods. Then after graduations, I thought that it might be good to actually trying to go to international universities to find out how can I actually bloom all the other approaches that are newly actually introduced to the research area. So I was granted one of the Australian international scholarships, which is focused on the talented students, came here to Australia, and then started my PhD in University of New South Wales, focusing on the bacterial genomics of Bordetella pertussis. This is one of the respiratory infections in infants causing whooping cough. So during my thesis, I did lots of bacterial genomics, whole genome sequencings, mouse studies to see how Australian epidemic isolates can actually fit into the populations. After that, I started a couple of postdoc studies and then joining from bacterial genomics to a labs who were focusing on microbiome studies, using my bioinformatics skills to investigate the microbiome populations in cancerous patients and also in healthy patients. This is how I drove from bacterial genomics to human microbiome, and then when I joined to CSIRO, I started focusing on the healthy microbiomes and how we can promote resilient microbiome.
What a fascinating intellectual and personal journey.
Thank you.
And you have such diverse experiences from Iran to Australia and working in multidisciplinary
That's
teams.
Right.
What does multidisciplinarity when you work in a team add to the research?
So I should say when you're working in like the multidisciplinary team with different cultural backgrounds from diverse countries that are joining together to solve some of the problems that are related to human health, you can actually negotiate well because everyone has like different perspectives that can actually bring it to like the brainstorming when you want to actually start designing the study, when you want to do some research, and then they have all different like knowledge backgrounds from microbiology to computational biology to like biomolecular studies, and this can actually help to actually design the study in a better way that can actually help you to have better results and outcomes.
Azadeh, I'd like to get into your research in the gut microbiome. What is the gut microbiome and why is it so important?
So let's start about what is microbiome, as David asked. So the human gastrointestinal tract, which starts from oral to our colon, is a home of trillions of diverse communities of microorganisms, including prokaryotic and eukaryotic microorganisms like bacteria, viruses, fungi, archaea, and also other eukaryotic microorganisms. We call them microbiota, which is composition of these different microorganisms. And when they interact with each other, and then they produce some metabolites in the gut and then interact with host, we call it microbiome, which is like a composition of the microbiota, different microorganisms, and their byproducts and their interactions with host. It is pretty important in host, regardless of being human or animals or environment. So in humans, we are assuming gut microbiome as a key organ for host. So it's like having like the brain. Without brain, you can't do anything. And now without gut microbiome, you can't do anything as well. So the human gut microbiome has a range of beneficial impacts on human health. Some of the main impacts include maintaining the integrity and function of the mucosal barrier, promoting and modulating the host immune system against pathogens, metabolizing harmful substances and xenobiotics like antibiotics, medicines, and so on, and providing macronutrients and metabolites such as vitamins, amino acids, and short-chain fatty acids, which are actually the results of digesting insoluble fibers and other components in the gut.
Fantastic. What sort of stresses can negatively impact our gut microbiome?
So there are different types of stressors. We can divide them in endogenous stressors and exogenous stressors. So I should say in terms of the endogenous stressors, it's like the age. So when you're getting older, your microbiome compositions change. Is it a good or bad? It depends on other conditions. When you're actually having some underlying conditions like chronic disorders, metabolic disorders, it might affect your gut microbiome composition. But the main strong stressors are exogenous stressors, including diet, travel, environment, lifestyle, and most importantly, medications and especially antibiotics, which actually targeting a vast majority of like the microorganisms, including bacteria, and then they can actually kill them during like preventing infections or curing like and treating infections in host.
When you mention antibiotics, we know they are very an important tool for fighting off diseases. But what impact do they have on these non-target bacteria, such as those good or beneficial bacteria in the gut? And why is our gut stressed after antibiotics? And is there also a quantity of antibiotics, or even the single dose can affect our microbiome?
That's a great question, Nina. So I should say that antibiotics, even one single dose, can actually distress and perturb our gut microbiome, and not only gut microbiome, but even other microbiomes in our body and organs, because their actually analytes of the antibiotics can actually circulate throughout our body and then impacted other parts of the microbiomes which are on the skin or oral or other organs. So why antibiotics are important? Antibiotics actually works as a double-edged sword. While preventing infections or treating the infections, which is a good indication for human health, they can also be like very negative impact on gut microbiome. They can actually kill beneficial bacteria because the mechanisms of the antibiotics that works on the most of the bacteria are the same, regardless of it is pathogenistic or beneficial. When you lose your beneficial bacteria, then the gut microbiome actually cannot work perfectly and then doing what it is supposed to do, including like providing vitamins, amino acids, or short-chain fatty acids. It also, when you lack beneficial bacteria, then the pathogenistic bacteria or opportunistic bacteria can colonize better in your gut. The other impacts of the antibiotic is that when you're actually taking antibiotics for a long time, then you will actually increase the abundance of antimicrobial resistant bacteria, or we call them multi-drug resistant bacteria. So when you have like these types of bacteria in your gut, which are colonized in your gut, then in the next type of administrating antibiotics, they might not actually easily respond to the antibiotic and treatments and cause some difficulties in host. It is like important for clinicians, policymakers, and even economics and governments that taking different types of antibiotics may cost for health and also like the financial problems that might actually issues with the underlying conditions and future actual impacts of the antibiotics. So when you take antibiotics, it might take 3 months to more than a year your gut microbiome recover. And then if patients have underlying conditions, then these dysbiosis and perturbations may go to like the stable dysbiosis phase, which actually might cause more problems in host. I can include several examples of how this might actually affect human health if I have enough time.
What a fascinating, multidimensional impact which antibiotics can have. What about the lifestyle factors? Are there any lifestyle factors that can, on the other hand, positively impact the gut microbiome?
Good point. Yes, sure. We do have lifestyle patterns that actually can negatively or positively impact our gut microbiome. So having too much alcohols or like different types of drugs might actually negatively impact the gut microbiome. On the other hand, having like good and enriched diet might improve positively your gut microbiome. The other parts of your lifestyle is exercise and sleep. Because gut microbiome has access and interactions with the brain, it can actually cause and correlate with the behavior. So if you don't have like good sleep, it might affect your gut microbiome. If your gut microbiome dysbiosis, it might affect your sleep. If you have good exercise, it might actually improve and positively on your gut microbiome composition, especially increasing the abundance of beneficial bacteria like butyrate-producing bacteria. But if you don't have enough exercise, then your gut microbiome might actually negatively impact it, and then you're losing butyrate-producing bacteria. So lifestyle is one of the main important stressors and factors on the gut microbiome, positively and negatively.
Azadeh, in your research, do you see any relationship between the impact on the gut microbiome and specific types of antibiotics? And what about the amount of antibiotics taken and the length of time taken?
Yeah, really good question, David. So in our recent publication, which is like a review paper about the dysbiosis of the human gut microbiome post-antibiotic therapy, we tried to list a couple of negative impacts of different types of antibiotic classes on gut microbiome composition. For instance, harmful and pathogenistic bacteria like E. coli or Enterobacteriaceae, and then reducing the beneficial bacteria like Bifidobacterium and Lactobacillus. Also, the duration of taking antibiotics is important. If you are actually in a condition that you have to actually take antibiotics for a long time, more than 1 month or so on, then it might actually delay the recovery of the gut microbiome to more than 2 years. New studies even shows that, for instance, in patients who had the tuberculosis and had to take antibiotics for a long time, their gut microbiome might not recover to its baseline fully, which might actually impact the function of the microbiome and impacting the human health.
Azadeh, what are the characteristics of a healthy microbiome?
So, really great question, David. So, although we have lots of research and studies about gut microbiome, about the microbiome, one of the main things that is still challenging is definition of healthy microbiome. So, because we are still discussing about how we can actually define healthy microbiomes, it is pretty challenging for most of the researchers in this field. But based on the current definitions, a healthy gut microbiome is characterized by greater microbial diversity and richness, a greater abundance of short-chain fatty acid producing bacteria. I should say short-chain fatty acid components are including butyrate, propionate, acetate, which are actually impacting the human health, including improving the immunity response in the body, and also enriching a metabolic functioning and a stable balanced microbiota composition. So, two of the main characteristics of healthy gut microbiomes are resistance and resilience. What are these two terms? So, let me just try to define them in a simple way. So, resilience of the microbiome is the ability of the microbiota to persist or recover from disturbances and stressors while maintaining its structure and function. So, as I said, microbiome has got a key role in producing vitamins, macronutrients, and so on. But when your microbiome got perturbed, your body still needs these macronutrients and vitamins. So the resilient microbiome trying to actually adapt itself under the new conditions, under the pressure of the stressors and perturbations, and trying to do functionally as good as what we have before these perturbations. So this is one of the main characteristics of healthy gut microbiome, which many researchers trying to see how we can improve it, how we can detect it, and how we can predict it when we want to actually administrating antibiotics, doing chemotherapy in cancerous patients, or even when someone wants to travel, which, as you know, travel is one of the main factors that might actually impact your gut microbiome, because we have travelers diarrhea, that someone's are more actually suspected to traveling when they are traveling from one country to another, or even one city to another city. Their actually gut might actually impacted. And we are trying to see how we can resolve these issues by improving like the resilience of gut microbiome to keep its function while it's disturbed.
So why do we want to model the impacts of stresses on our gut microbiome?
Yeah, so why it is important to model that? Because we want to find out if someone is going to take antibiotics, for instance, what's going to be like the impacts of these dysbiosis. Is this individual might have stable dysbiosis that might impact like gut organ access, including like gut-brain access, gut-liver access, gut immune access, or the individual can actually recover this from these situations and its microbiome actually recover faster. So if we can actually resolve these problems, we can help clinicians to find out how they can prescribe these types of medications to individuals. Can they actually give them some interventions that prevent a high actually impact perturbations or strong like stressors to actually affect less on the gut microbiome? So this is pretty important by modeling that, because if you have enough data to do so, you can actually help clinicians to help patients to recover faster and they're not going to like the stable dysbiosis phase.
It seems like a lot of education is on the way for
That's
a
right.
range of practitioners, which is a good thing.
Yeah.
Currently, is there any treatment that can potentially minimize the impact on the gut microbiome when antibiotics must be taken?
Good points. It's really good point, Nina. There are lots of researchers and research teams that are trying to investigate how we can actually recover the dysbiosis microbiome faster or prevent the impacts of dysbiosis microbiome on health. So many strategies has been suggested. One of the main things that we can actually talk about here is that before taking antibiotics, you need to have like high diverse microbiome. So how you can do that? By having like a good lifestyle, having exercise, reducing your alcohol takings during the days or weeks, doing like sleeping well, and also taking some probiotics, prebiotics, and also high fiber diets that can actually promote the richness of your microbiome and then increasing the beneficial bacteria in your gut. But what happens when we actually taking antibiotics? Anyway, antibiotics can kill all these beneficial bacteria. Again, by using some of the interventions like probiotics, prebiotics, high fiber diets, and also if we have like more complicated issues like fecal like transformation or transplantation, we can actually help patients to recover faster. Some studies shows that probiotics might not actually work well in terms of recovering the dysbiosis, because as soon as actually they go to like the colon or like distal intestine sections, they might actually compete with like the microbiomes itself, and then the colonization might actually cause delays in the recovery. So new studies shows that if we actually combine these probiotics with other supplementations, we can actually give like promising results and outcomes by fasting like the recovery of the gut microbiome.
Your research aims to develop a resilience model. How will you see this resilience model be used to benefit patients, and how will you see it being used in doctor surgeries?
So let's talk about my project in MOSH and CSIRO. So we focused on improving the resilience of gut microbiome, how we can actually increase the resilience, how we can detect it, and how we can predict it, because each of them might actually help individuals and clinicians and also policymakers to have and decide like the better approach in terms of the reducing the impact of gut dysbiosis. So In my projects, we try to define a couple of biomarkers that can be used for detection and prediction of the resilience in individuals. It is pretty difficult field because we are still struggling about what is the resilient gut microbiome is and how we can actually measure that. Measuring a resilient gut microbiome is pretty difficult because the microbiome is a very complex community. It has interactions with hosts, and it needs longitudinal studies and lots of data, because if you want to use like the statistical approaches, machine learning approaches, artificial intelligence approaches, you need high actually accurate data to be used to model the resilience and find out so how we can predict this in individuals. At the end, I should say, if we can actually measure the resilience and define some biomarkers that can help us to model and predict the resilience, it will help clinicians for and also policymakers for precision treatments, precision probiotics, and then personalized treatments for like different types of individuals in different types of conditions, like for example, patients that have cancers, patients that have some underlying conditions like diabetes or metabolic disorders or liver disorders. So either of these conditions might actually need specific type of interventions to help the recovery of the gut microbiome or to increase the resilience of the gut microbiome. These modelings can help the clinicians and policymakers for personalized medicines, personalized probiotic therapies, and help us to actually reduce the time and the impact of the diseases.
One aspect which I found very, very interesting, Azadeh, in your research is the gut-organ axis. And I wanted to ask you about your cancer research. Is that part of that sort of strain of research, gut-organ axis, and how does microbiome impact cancers? What does your research say about that?
Yeah, great question. So one of the studies that I've involved is like the impact of gut microbiome in like cancerous patients, especially in the colorectal cancers and also liver disorders. I'm a member of a team from UNSW and Microbiome Research Center that are investigating the biomarkers of the different types of liver disorders, from cirrhosis to liver cancer, and how we can actually use these biomarkers or multi-omics biomarkers to differentiate these things and also predict the progress of the disorder. Whether based on these biomarkers, we can predict the outcomes that can go to the latest stage of the cancer, or it can actually improve the situations and conditions in individuals. So as you know, and I said previously, different types of bacterial taxa can actually be introduced as like the main cause of specific like disorders. For instance, many studies shows that there are correlations between Salmonella typhi and gallbladder cancers, or Helicobacter pylori with gastric cancers, and even like specific bacteria with like breast cancer, which is not completely related to the gut, but it can actually impact like the gut. We did have like a review paper about like non-invasive biomarkers that can help us to predict and detect like early phases of the breast cancer. So microbiome is one of the omics or multi-omics next to like the genomics, transcriptomics, proteomics, and metabolomics that can help us to define a panel of biomarkers for detection of earliest stage of breast cancers, and also how we can actually predict like the progress of the disease in patients. Same as this, in the liver disorders, the team tried to actually define a panel of biomarkers from like the cytokines and immune systems, from metabolites of plasma, from like lipid plasma metabolites, and also microbiomes from oral microbiomes and stool microbiomes, to see if they can actually define a panel of these metabolites and bacterial taxa that can help us to predict and identify and classify different types of liver disorders.
Azadeh, I'm so fascinated with the comments and research about travel. And you mentioned that when you travel from one city to another, that can affect gut microbiome. Would you mind sort of explaining to an average listener, how is this possible? Let's say we go from Perth to Sydney and vice versa. How can our gut microbiome change in the same country, but obviously on a different side of Australia, for example?
Yeah, that's really fascinating to actually talk about these like types of travelers' diarrhea. You know that we all experience that when we actually travel from city to a city, we always say that the changes in the water have actually impacted us, or we have like some vomitings or diarrhea, or even in kids we do have some more complicated conditions. And the recent studies shows that there is like strong correlations between the profile and composition of your microbiome and how you actually respond to these type of conditions when you travel. So traveler's diarrhea is a common gastrointestinal condition that affects travelers visiting regions with poor sanitation or contaminated foods and waters. It's primarily caused by ingesting pathogenic bacteria, viruses, or parasites that disrupt the gut microbiome. But if your gut microbiome is not diverse enough, they might actually impact you more. If your gut microbiome is diverse enough, then its function can actually cover the dysbiosis and perturbations during the traveling. So that's the importance of having higher resilient microbiome, which can actually adapt itself in different conditions and environments. So based on the research studies and new studies, it shows that, for instance, Faecalibacterium pearson, which is one of the short-chain fatty acid producers, might actually decrease during the travels based on the individual's actually conditions, and then cause like a couple of underlying situations during the travels. The other factor is that you might actually get sick during like the travel, especially if you are actually traveling using like train or public transportations or something like this, that you might actually take like different types of respiratory infections, and you have to take antibiotics. Taking antibiotics then have all other actually perturbations and impacts on the gut microbiome. And how we can actually try to recover and improve these situations while traveling is trying to steps to restore our gut health, taking probiotics and prebiotics, hydrating, which actually helps gut microbiome to replenish lost fluids and electrolytes, and giving more electrolytes and good source of nutrients to the microbiome, using fermented foods like yogurt, kefir, kimchi, and these sorts of like foods, and avoid accessing antibiotics. So if you have like viral respiratory infections, taking antibiotics can't affect that. So like, yeah, bearing in mind that if you're traveling, trying to prepare yourself for these kind of conditions, and then think about that before start to travel.
Absolutely fascinating. Azadeh, thank you very much for this extensive interview. We would love to have you back in the future. And all the best in your future research.
Thank you. Thanks a lot for having me here. And then, yeah, bye. Sorry.